Welcome everyone to the very first longevity live stream. During this stream, we're going to go through ways to extend health span and answer any questions that you may have. And I'm stoked to have Eleanor Sheekey from the Sheekey Science Show on with us today. So welcome, great to have you here.
Hey, thank you for having me on and being your first live stream guest. It's kind of a privilege.
Yeah, cool. So I think one of the things that I've really been pushing on this channel recently is, supplements are interesting, but really it's diet, exercise, sleep, meditation, other things that will really help to extend health span. So I'm just curious at the moment, what is the diet that you're using and what's the things that are exciting you in that area of research at the moment?
So I guess diets, I still think I vary quite a bit in terms of what I eat, but mainly it's, I guess, mainly plant-based. I'm kind of a pescatarian, so I tend to eat fish, not meat. A lot of whole foods, vegetables. I'm not so big on fruits, but that's just more of a personal preference. I think as well, food timing is something I tend to find more interesting than necessarily food type. I'm sure like viewers have heard of fasting and time restricted fasting. And there was also some more recent data about caloric restriction. I don't try to count calories or anything, so I have no idea if I'm eating more or less than I should be. But as long as I don't feel like I'm overindulging, and I combine that with exercise and just do what my body feels comfortable with, that's kind of where I'm at with my diet. Is not particularly too interesting.
Yeah, the stuff around intermittent fasting I find quite interesting because towards the beginning of 2021, there was a large Cochrane review that looked at intermittent fasting, and when you calorie match the diets, there didn't seem to be much of a difference. Because I think, one of the big things that we were excited about or hoping for is that the mice data, where if you fast the mice for 16 hours and they can only eat within that 8-hour window in the day, we were hoping that those benefits that those mice were seeing would translate into humans. But from this Cochrane review and from all of the studies that I've looked at, it doesn't look like humans actually get any further benefit if you simply just lower your calories and match them. So yeah, I mean that's personally why I don't intermittent fast, aside from the 5 to 7 day fast that I do roughly every 3 months, because I think it's those prolonged fasts that actually have got a chance of improving health span, because that's where you're really activating autophagy. So I'm curious, do you fast at all, or are you just waiting for more data to come through?
Yeah, no. So to add to your point, you're right. That's to enter like ketosis. It does take a long period of time for you to deplete the glycogen stores that you already have present before the metabolism switches over. And so, I think there's different reasons for why there isn't. I haven't looked at that study in particular, but I think part of it's the power in numbers and how any kind of diet analysis in humans is always going to be confounded by multiple different variables. And so that's probably one of the issues why maybe there hasn't been anything particularly promising coming from these studies. On from that point, I think part of the reason why I'd say I tried to intermittent fast is more I just don't like eating too soon to bed because I sleep better. And so I think that's something that may have benefits. But then, going back to the point about fasting, I've never done like a full beyond like 24-hour fast, partly because I like food, but also I think at the moment there are other things like exercise that I do already and try to, in terms of food quality, as long as that's okay. But for me, I guess I'm more worried about like menstruation and other factors, like especially for a young, I guess still growing female. I think I have no real need to at the moment. It's from my side. Obviously, like there is like emerging data supporting these like longer term fasts. Recently, there was another update with the calorie trial that was, I can't remember when it was done. It was done a couple of years ago, but obviously lots of different measurements were taken. And I think they had some human participants, and on average, they were doing about, you might have read this as well, it's about 15% reduction in calorie intake they've managed in total. And they've now looked at different measurements, and the most recent paper looked at the thymus. Is that right? I can't quite remember.
Yeah, it's interesting because The patients that I see in the clinic, so particularly my overweight patients who want to lose weight and want to lead healthy lives, it's incredibly difficult to make a lasting change. So yes, they might be able to lose weight, over a 3-month period, and that's great. But if you have a look at the 2-year period, that's when it's quite difficult to actually maintain that weight loss. And that's where medication, so the diabetic medications, GLP-1 receptor agonists, those seem to be very powerful for weight loss. And I've been prescribing quite a lot of those in the clinic because it seems that that's how you significantly reduce the feelings of hunger, and therefore you can actually start to calorie restrict yourself if you like, and that's how these drugs cause weight loss. So I think it's going to be quite interesting to see, whether we can start to use those medications so that people can actually lead those healthier lives and actually give them a fighting chance to reduce their calorie intake over a multi-year period. So I'm very curious to see if that's a valid way of keeping the calories down, because that calorie trial is very interesting. And I think the thing I like about it is that it does seem to confirm what we're seeing from the mice data. Um, mm. But
Yeah.
aside from the fasting data that we've kind of talked about, is there anything else in particular that you're doing with your diet? So, for example, the keto diet is very popular. I'm less convinced about the data, but I'm curious to hear your thoughts.
Yeah, I mean, there was a very nice review article that came out at the end of last year, I believe, that kind of summarized an up-to-date overview of all the different mouse studies and other model organisms looking at these different diets. And I think, yeah, you're right, that if you looked in terms of like anti-aging potential, like none of the diets have really have much support at the moment besides like fasting, which has mainly come from this calorie trial. I think if I looked at my diet, someone could probably say, yeah, she seems to have a mainly ketogenic diet. But I think again, that's just what my body likes, and it's not because I've seen a particular study and gone, okay, the science looks good. That's why I'm doing it. It's more what my body feels best, and I think most people can tell if they feel like they're functioning as a good, feel awake, feel alert, feel that they can focus. And yeah, I eat a lot of nuts, which I guess is like a kind of classic keto component. So yeah, so basically I think there isn't enough data to really, still to this point, to be certain what is the best diet to follow. And I think again, there will be some personalized element to it that is somewhat being explored at the moment with all these genomics approaches. But yeah, besides that, I think it's a matter of just waiting for more data to come out.
Yeah, I think there was a lot of excitement around the carbohydrate insulin model of obesity, where the theory was that it was mainly high levels of insulin that was causing people to feel hungry, and therefore they would eat more, and it would just spiral out of control. But all of the data that I've looked at when on exploring that theory, it doesn't really seem to stack up, and I've not seen that in the clinic. If when I encourage my patients to have less carbohydrates, I've not really found that that's worked in my clinical experience. But what does seem to work is if I encourage them to have whole foods, and that does still include carbohydrates. So if they, and instead of having Fruit Loops in the morning, if they switch that into porridge with no added sweeteners and whole blueberries and those sorts of things, that actually seems to make a difference, because it's the fiber content that has a crucial role to play in the feedback mechanisms from the gut, that's a way for actually tricking your body, if you like, to having less calories in. So I think that the point of personalization is so crucial, that any diet or any intervention, it needs to work in with someone's lifestyle. So, say for example, if I've got someone from a particular culture and they always eat a particular food set, if I then suggest to them, oh, you shouldn't be having that food, that's not going to work. But if we can find ways to actually make that food healthier, so increase the fiber, increase the whole food intake, that's actually got a chance of working. And all of if you have a look at the weight loss data for lifestyle interventions over a 2-year period, it's incredibly difficult for patients to actually lose that weight and then keep that weight off. And that's why I get quite passionate about, it's not about necessarily blaming people for being overweight. Because I think once you get to that overweight stage, it's then incredibly difficult to get it off and keep it off. And that's where I use medications to help shift things, to actually give patients a fighting chance.
Yeah, you made me think of two
Yeah.
quick things there. So one, I was fortunate enough in my master's degree to have some lectures by Giles Yu, who is a geneticist at Cambridge University looking at obesity. He has a couple of books he's written. And one of them is he very much argues that obesity isn't necessarily a choice and that there is some genetics components to it. And you're right about how it's hard to get out of that kind of feedback loop in terms of losing the weight initially. But the second thing that you made me think of is there's been, at least of people that I know, a lot of interest in people taking, I don't know what the equivalent is in New Zealand, but there's something called like Huel in the UK. It's some kind of like powdered food source. So instead of like having a proper like nuts or some actual vegetables, you have like just a shake or something that has all of the vitamins and everything you need in it. And I've not ever tried these things, but I guess my one concern is, what would be your opinions on these kind of, so like they claim that this is sufficient for your food, but if there's not the same like fiber content or like lack of microbes that would be beneficial for the gut, I mean, have you seen any data in terms of the effects of people following these kind of diets, the powdered foods?
I think it all depends on what sort of life you want to lead. Do you really want to be a person who's just eating these powdered foods for the rest of your life? And that's the thing that I try and talk about in the clinic. Any change that's made, it needs to be something that you can actually sustain in the long term. So yes, you might be able to do these powdered drinks or meals for 3-6 months, even a year, but do you want to be doing it for 40 or 50 years? I'm not sure. So that's where I think it really comes down to using whole foods. And on the point of obesity is not a choice, it's very interesting that you say that. One of the classes of medications that I frequently prescribe in the clinic is SGLT2 inhibitors. So they're a class of type 2 diabetic medication where essentially it makes the body pee out sugar. And it's very effective for reducing blood sugar levels, but it's also quite effective at weight loss. And how that medication works, it simply blocks one of the transporters in the kidney to make the kidney pee out that sugar. So you can imagine there are some people who have got, they've got heaps of these transporters that hold on to sugar in their body, and other people that don't have many of these transporters, and therefore they can pee out that sugar. So just that one example is a classic case where some people they've got the genes for obesity, and someone else doesn't. It's the same thing with uric acid. So some people have got a lot of transporters within their kidney to hold on to uric acid, and other people don't. And that's why some people develop gout and others don't. So yes, there are lifestyle things that will increase your chances of having gout flares, but at the end of the day, it is in your genes, and I think it's unfortunate if people are ridiculed for being overweight. I mean, yes, there are exceptions. Yes, some people are just having lots of sugar in their diet and they're eating to excess and they're not exercising. Then yes, sure, we need to do something about that. But for a lot of overweight people that I see in the clinic, they've tried so many things. And I think it's awesome now that we've actually got interventions that can work for people and that can extend their health span.
Yeah, and I mean, this has taken me back a couple of years to when I knew a lot more about this topic, because so Giles, when he lectured us, as I said, he studies the genetics underpinning obesity. And so there's also a lot of like neuronal receptors that also are involved in the leptin, ghrelin signaling, and how obviously, yeah, there's communication between like your gut and your brain in terms of like triggering your hunger or when you're full, etc. And so even slight genetic changes in these different receptors affects like how the affinity for the different molecules to bind to the receptors, and obviously the downstream signaling processes. And so based on these identifying those different genes and the proteins they encode, you can develop new drugs to target them. And so, yeah, there definitely seems like a lot of promise in this direction.
And it's just cool because I can see those effects in the clinic where I start prescribing these medications alongside a great diet and regular exercise. And people lose the weight, they keep it off, and they're feeling great. And it's you do get into this positive feedback loop where if something is working, you're going to continue doing it. So that's just been my clinical experience. But moving on from sort of diets and things, I'm curious to hear about the exercise that you do, because I've just done a video that's due to be released, I think this time next week, on running. So I'm curious to hear your thoughts about exercise.
Yeah, so I mean, in terms of what I do, I mean, I cycle to work pretty much every day. I'm always in the lab, even on a weekend. Um, so that's like a solid 30 minutes in total, probably each day. And then I run as well. I'm like not like short distance, but not like marathons. I mean, I do like a between 5 to 10K kind of casual. It's more for me, it's like a stress relief to go running as opposed to making sure I'm exercising. So I see that as the kind of beneficial part from the exercise as well. And then obviously I know resistance training is important. And so I guess like I don't really go to the gym, but I do like maybe push-ups, etc., just to try and get some strength. But yeah, otherwise I'm quite a cardio person, but that's again just more of a personal choice than necessarily because because it's I mean, also HIIT exercises is something I think we've both spoken about before when we've been on each other's channels. There's also there's a lot of evidence, right? At least in mice, that obviously exercise is beneficial for neurogenesis, which is something that's I think many people are would care a lot about when they age, is their brain operating as it would usually be, like memory consolidation, just interactions, learning. Like the brain is so important to preserve, and so if there's any kind of benefits coming from exercise, I think that's one of the main ones.
Yeah, I think in terms of longevity, having a mixture of cardio and resistance training, I think is just so crucial. Again, just coming to my clinical experience, patients that I treat in the rest homes or who need private hospitals, it's unfortunate because you can see that they might be overweight, but in terms of their muscle content, it's so low, and they just don't have the strength to look after themselves anymore. And that's where resistance training can really improve things, where you can hopefully hold on to your muscle strength as much as possible. So the types of exercise that I do, I make sure to exercise roughly 5 times a week, and I do a mixture of cardio and resistance training. Obviously, it's been quite tricky during COVID times to get into the gym and things, but so I've started to set up a home gym here. But in terms of the cardio that I do, since time is limited, I try and make sure to do a high intensity interval workout, because I'm trying to get as much benefit as I possibly can in that short period of time. So, sometimes I've only got 10 minutes to do an exercycle workout, but if you do the sprint workouts in that 10 minutes, you can actually get a lot of benefit within that 10-minute window. So that's personally the exercise that I've been doing, the mixture of both cardio and resistance training. Yeah. Is there anything else in terms of exercise that you've read recently?
I guess just to go on from your point, obviously, I remember recently I read some stuff about selenium and how that also had neurogenesis benefits. And the reason why I guess it's interesting to follow up on these studies is, as you say, there are some reasons why people can't exercise and get the benefits from exercise. And then you end up in like a negative spiral where if you can't exercise, you don't get the benefits, and then you progress the whole situation gets worse. And so finding alternative ways of maybe mimicking the benefits of exercise. Obviously, I don't think you can truly beat doing the exercise yourself, but for those who can't do the exercise, it's important to do these studies. But yeah, I don't think I've really read anything. Yeah, you probably know more than I do. If you've got a video coming out, yeah, it's been a while since I read any exercise-focused papers.
Yeah, I think on the point of exercise and not necessarily getting those benefits or as much benefit, that's one of the big, so I'm just going to use this as a segue into Rapamycin. That's one of the big reasons why I'm quite passionate about the Rapamycin trial that I'm doing, because older people, exercise is fantastic, but they don't get as much benefit compared to when they were younger. And I'm hoping that if we combine intermittent rapamycin, once a week, and the other times we exercise, I'm hoping that that will give even greater muscle performance benefits compared to just exercise alone. So yeah, I'm quite passionate about that because I see this every day that as people get older, they get weaker and weaker until the point where they can't look after themselves. So just to give people an update about where we're at with the rapamycin study. So VitaDAO is currently evaluating the project to see if it's something that they're going to help fund. So that's very exciting. We've recently submitted the trial. So when we're running, just for the audience, when we're running clinical trials, one of the things that we always should be doing is registering it with a clinical trial registry, because that way you can actually you can say what your design is going to be, and you can say what you're going to measure, and what the primary outcome is going to be, because that way there's no trickery that can really go on, because you've said what you're going to do and then you're going to test it. You're not going to try and do what's called p-value hacking. So we've just submitted our trial design to the clinical trial registry, and once that's done, and once we've got IRB or ethics approval, which will hopefully be in a couple of months from now, 2 or 3 months, then we'll be good to go. So it's quite exciting. There's still a little bit of funding that we need to get, but VitaDAO will certainly be helping. Well, I'm hoping they'll help with that as well. So yeah, that's where we're up to.
That's really cool to hear. Wait, you have to remind me, how big is the trial going to be, and what was the duration of time?
Yeah, so with this trial, it's a phase 2 study. So what we mean by that is primarily it's going to be a safety trial. So in total, there's going to be 40 people. So 20 people taking the rapamycin and 20 people taking the matching placebo. Both groups will be exercising as well. And so this study is, there's no studies that I'm aware of or that I've come across in all of the research that actually combines exercise with mTOR inhibitors, which is what rapamycin is. So we need to first make sure that this is a safe thing to do, and we're hoping to see a signal in the data that it's actually a good thing to do. Because if we can show that it's both safe and that it's that it might be effective, then that gives us even more justification to the IRB and ethics approval committees that we should be doing a much larger study to actually see if there's going to be a positive benefit. So, in terms of me being able to prescribe rapamycin to older people, we're still a few years away, but this is the start of actually getting it done. That again, showing that exercise combined with rapamycin is both safe and potentially a good thing to do. So yeah, that's the kind of phases that we need to go through.
Cool. Yeah. In terms of rapamycin, because obviously, I guess some of the evidence, the strongest evidence for it's come from the ITP study, which I know you've probably more than I do about it, but was the fact that even giving rapamycin to mice at a late life stage enhanced their lifespan. And interestingly, I came across it was a preprint, so it's not actually gone through peer review yet, but looking at the other stage of life, so very early development, they gave rapamycin to mice for the first 3 to 4 months of their life, and that also extended their healthspan and lifespan. And so I thought that was a very interesting study, because it kind of highlights how sensitive the early developmental time period is to these different therapeutics.
Yeah, I think it is very interesting that rapamycin seems to give these lifespan extension benefits in mice in their older years, and it will be quite cool if we can see that in humans. But trying to design a study showing a lifespan extension in humans, we'll be waiting decades for this, and that's why I think if we had to choose anything for or any reason for why rapamycin should be used in older adults, I think it's the exercise. I think it's such a compelling reason, and that if we can find a safe way for older people to hold on to their muscle strength, I think that's pretty powerful. And even if you have a look at their resistance to disease, so against pneumonia and things like that, you still get significant benefits in older adults if they're stronger and if they're less frail. So yeah, I'm hopeful that with this study that I'm proposing, and with the Pearl study, which is also another rapamycin study, I'm hoping that the tide will change, because in medicine, there is a little bit of stigma against rapamycin because it's primarily used in patients that have had kidney transplants. So to make sure that the body doesn't reject that organ. So if so the fact that we might be using, a medication that lowers the immune system to extend or to improve muscle performance is slightly baffling. But I think people just have to realize that it's not necessarily the rapamycin that's causing that reduced immune response. It's the combination of the other medications as well that's given in those kidney transplant patients. And actually, on that point, there was, I think it was published in 2017. There was a study that looked at mTOR inhibitors. Again, that's what rapamycin is. And it wanted to look at the influenza vaccine to see, would there be a stronger response to the influenza vaccine if you used mTOR inhibitor as well. And there was less influenza disease, and you could also see that there were higher levels of the influenza antibodies within the body when those people were using mTOR inhibitors. So I think that's quite a powerful piece of evidence against the idea that these mTOR inhibitors would worsen the immune system. If anything, I think it probably would increase or improve things. But further research will tell.
Yeah, I guess that's the thing. So mTOR, I guess primarily people talk about it in terms of blunting cell growth, but I guess it kind of is one of those proteins that seems to just be linked to so many different molecular signaling pathways, and so it's kind of like one of these central hubs in a cell. That sure, cell growth is just one component, but it also is all sorts of inflammatory signaling overlap as well, and so I think, I think that's possibly why there's been some conflicting data. It's just I don't think we understand fully what mTOR does in a cell. I think that's still something that's worth exploring at the molecular level as well.
Just to segue, Matthew from the comment section has just asked about epigenetic reprogramming. And I know you've published a couple of videos about this, and there's been a couple of interesting papers that have come through. So do you want to talk to us about epigenetic reprogramming and what you think about this field as a whole?
Yeah, sure. So I guess the first place to start is what does one mean by reprogramming? And so I guess, we all have different cell types. I think there's about 200 different cell types in the body. And a skin cell was very different to a liver cell. And despite having exactly the same DNA, they're doing very different things. And ultimately, that comes down to the fact that they're expressing different genes. So that's just the premise of differentiation, having different cell types. And so reprogramming is basically the idea of turning one cell type into a different type of cell. And cellular reprogramming is more commonly referred to the context of taking a differentiated cell type and turning it back to a stem cell. And so it's you change the cell's identity. And so then the question, which comes back to epigenetics, is how come if the cell's got the same DNA, how is it expressing different genes? And that comes down to the chromatin and the epigenetic marks on the different histone proteins that package DNA into the nucleus, and also to some extent the DNA methylation marks, and how the packaging differs in different regions of the genome dictates which genes are accessible to the protein factors in a nucleus that can cause genes to be expressed. That was probably not the best explanation, but basically epigenetic marks and chromatin structure is really important for dictating gene expression and ultimately what a cell is, its identity. And so going from one cell type to a stem cell, you have these changes in epigenetic marks and chromatin structure. So stem cells in particular have quite large nuclei. They seem to be more less heterochromatic, so they're more open, they're more accessible for gene expression. And so the question is, how can you cause one cell to go from a very compacted state to this more reprogrammed stem cell open chromatin state? And there's lots of different strategies that are kind of being investigated. The main one that kind of kickstarted the field was the discovery of the Yamanaka factors. So 4 different transcription factors that, if they're expressed in a cell, can cause that cell to basically effectively go from a differentiated cell to a stem cell-like state. And in that process, the epigenome is also altered. And do you want me to keep talking? I've been talking for ages now.
Essentially, what I take from that is that if you've got a skin cell that's and you want to turn that back into a stem cell, you can do that, and that's so the epigenetics, it essentially tells the cell what it's going to be. So whether it's going to be a skin cell, whether it's going to be a kidney cell or a liver cell, and the epigenetic reprogramming is hoping to try and reset or get that cell back to a more youthful state. I think that's the overall hope. But the thing that's kind of confused me, the Yamanaka factors, they've been known for a while. We've known about these factors and how to use them to turn a skin cell or a kidney cell back into a stem cell. But now there seems to be quite the push in some research centers that if we only use a little bit of the Yamanaka factors, that we can then rejuvenate the cells and make them into a more youthful state. And I suppose that the thing that, and I recently had Dr. Morgan Levine on the channel from Altos Labs, who's looking into epigenetic reprogramming. One of the things that she was talking about is that she really doesn't think that we should be converting cells back into stem cells. Ideally, we just want them to be a more youthful state. And knowing which parts of the epigenome we should be influencing, that's crucial, because there are some points that seem to correlate with aging and that we probably want to target. But equally, there are other points in the epigenetic program that we probably don't want to be touching. And using the Yamanaka factors, to me, it just blanket says we need to reset everything. And with that can come a lot of problems. And I've just been reading from some of the commentary with the latest papers that have come out that have used the Yamanaka factors to try and reset cell aging. And some of the commentary is positive, but other commentary is saying, well, hang on, we probably don't want to be doing this because we don't know if it's safe, as in will it cause, cancers and things, because you don't want a kidney cell to turn into a skin cell, for example. So I'm curious to hear your thoughts around, why the hype around the Yamanaka factors has re-kicked into gear when we've known about the Yamanaka factors for quite some time, and the idea of using them to partially reprogram our cells.
Yeah. So obviously that was, I think it was 2009 when the first Yamanaka paper came out, and then obviously from then there was a lot of initial excitement. And I guess what happened was they first did it in mice, and then they had them overexpress these factors, and that caused the formation of these so-called teratomas, these kind of horrible, gets me the creeps just thinking about them. You think tumors are bad, but teratomas are just tumors, but they're different bits of differentiated stuff. So you got bits of teeth and some of them, bits of hair, and it's just not good. So complete reprogramming has concerns in terms of uncontrolled proliferation and complete loss of cell identity. And so where things have gone more recently was a 2016 paper that did something called partial reprogramming, where they express the factors in mice for only a short time period, and then they leave it for a little bit, and then they express them a little bit again and leave them. And so the idea is by having a kind of partial expression of these factors, you don't get complete reversion to a stem cell state, but maybe somewhere on the journey between the two states. And that enables a bit of potential rejuvenation that's up for question, but it doesn't cause the cell to lose its identity. And so it might, help basically promote, different security factors, or might improve signaling within the cell. I think, again, that's kind of not clear exactly how the partial reprogramming is going to be beneficial, but it enables the cell to still know what it is, that it's not going to have any concerns in terms of tumor growth. And so I think the two questions are, one, again, in mice, the Yamanaka factors, the OSKM seems to be sufficient, but in humans, like at least currently, it seems that you need to have additional targets as well for more optimal reprogramming. Also, because one of the factors, MYC, is like an oncogene if that's overexpressed in many different cancer types, and so overexpressing that in human cells isn't something you don't really want to do. But actually, I think one of the more interesting advances I saw more recently was using CRISPR approaches to express the Yamanaka factors. And so currently in the mouse studies, they have genetically modified mice where they effectively are overexpressing these Yamanaka factors. So you have, like, in our cells, we have the genes for the different Yamanaka factors, and they're adding in additional copies and overexpressing it. And what this CRISPR-based approach did was actually, instead of adding in additional copies, they target the endogenous genes that already exist in the cell and activate those instead. And so by using the endogenous expression, it helps to retain like the regulatory information that's encoded in the genome. You're not overexpressing, so it might, in terms of dosage of the factors, it might be more beneficial to have a lower expression for a longer period than to have a higher expression for a shorter time period. And I guess as well with CRISPR, you can more easily multiplex if you wanted to target multiple different sites in the genome. And you could do that to maybe enhance the reprogramming process. So I don't know if I answered any of your questions, but I think, yeah, one of the main questions is how would you ever translate this to human therapeutics? And obviously the mouse studies, they have them modified in all of their cells. And obviously there's no way in a older human patient that you'd be able to do that. So if there's already kind of like not that amazing rejuvenative effects from the mouse studies, what would ever be seen in humans, I don't know. But definitely the translation is the big challenge at the moment, and identifying where the safe stopping point is in human cells compared to the mouse studies.
Yeah, it's just, for me, chatting with Dr. Morgan Levine, again at Altos Labs, it seems that there's some crucial questions that we don't know the answers to. We don't know why the epigenome all changes over time. So what's actually driving that? We don't necessarily know which parts of the epigenome we should actually be targeting. And because of those two unanswered questions, I don't know, I'm skeptical about blanket using the Yamanaka factors just to reset it, partially reset everything. It just seems like it's not a particularly nuanced approach. Because again, I would have thought if we can find out why one particular part of the epigenome is changing, and if we can change that back to a more youthful state, then we'll get X, Y, and Z benefits. It seems that then we could target a treatment just for that part of the epigenome. And that seems to make a lot of sense to me. And I think that's what Dr. Morgan Levine is looking at. So yeah, I think this field is very interesting, but I'm not yet too excited about using the Yamanaka factors.
Yeah.
Yeah.
I mean, the other thing to think about is the fact that, as I already said, our different cell types are different because they have different extents of chromatin compaction to modulate their gene expression. So expressing the same factors in these different cell types has differing effects. Like some cells are easier to reprogram than others. And so that's something else that you have to kind of bear in mind. I mean, that could be a good thing because maybe some cells you like, definitely like in the brain, I'd be very skeptical of any neurons going back to a more stem cell like state. But that might work in our favor, but at the same time, I think it's still a very early stage in terms of understanding the chromatin landscape and how that influences a cell's identity. It's cool. I mean, this is the kind of stuff that I find very interesting. Like senescent cells, which is what I study, have their own kind of interesting chromatin changes that are different from what's seen in like a normal healthy cell. And it definitely seems to be, especially with techniques like DNA methylation is one of them, but there's all sorts of improved methods to better characterize the chromatin landscape of a cell that's coming out. And I think that's also why there's been maybe a lag in expectation as to where's all these results coming from, but it's more because the basic biological understanding has been dragging behind a little bit because we've been catching up with these newer techniques to study it.
Yeah, I think that this field is very interesting, and I think I think it definitely needs to be done. I'm glad that money is starting to flow into this field. Um, and again, I keep referencing the conversation that I had with Dr. Morgan Levine, but it seems that these therapies, they're at least a 10 to 15 years away, and hopefully by that point we'll have a much clearer understanding about what is driving these changes, and what we should actually be trying to reverse, and then what those health benefits, if any, we will get. So yeah,
Yeah,
I think
you just
go
made
on.
me think of something else. So far we've been talking about like in vivo reprogramming, so expressing them like in the mice or if it ever went like human patients. But the alternative strategy is to obviously take like somatics, the differentiated cells from a patient, and ex vivo reprogram them for like use in like regenerative medicine approaches. So I think that's more likely like as a method to be seen before any in vivo approaches.
Yeah. Is there anything else you wanted to touch on with the reprogram? Otherwise, we can have a look at things like NAD precursors again, because that's always a topic that I quite enjoy talking about.
Yeah, I mean, I think it's been a lot of excitement around the field at the moment. Yeah, with things like Altos Labs coming out. I think it's kind of cool science. And I think we're just going to continue to see more papers come out in it. And I guess just a touch from, there's more than just the Yamanaka factors that have been linked with like potential rejuvenation approaches, but also like the heterochronic parabiosis and as like another way that may have rejuvenative effects, but without having a way of really defining what rejuvenation is, like molecular hallmarks that we can say, okay, this is definitely rejuvenation. It's hard to really validate that rejuvenation is occurring, if that makes sense.
Yeah, absolutely. So onto things that people are actually starting to use now, and I personally use, is NAD precursors. And from the last time that I chatted with you, am I correct in saying you don't use NMN or NR or niacin? Yeah, it's interesting because people like Dr. Matt Kaberlein, who's very very clued into this area of research, doesn't. Dr. Morgan Levine doesn't. So it's I think. But in saying that, there is interesting human data that's starting to come through around NAD precursors, specifically, this papers that came out around the recovery from COVID-19. So yeah, I'm curious to see what your latest thoughts are around using NMN and NR.
Yeah, I mean, so again, you're probably more clued into the more recent papers than I have. It's been a while since I've really done a deep dive into it, but I think just from my side, like NAD plus is just like a really important molecule within our cells. Like it's not just involved in metabolism, but also it's like a cofactor for different proteins. And so I think it's like undeniable that I'm sure if whatever precursor was somehow getting into the cells, into the different tissues, that it would be having some kind of impact, and therefore is likely, to be potentially beneficial. But I guess it comes, back to in terms of, is there like a kind of threshold, or is there any negative consequences of having too much NAD plus, or how does it have any like feedback mechanisms in a cell where it thinks it's got too much, so it shuts down other processes? I'm being kind of very vague, but that's partly because I haven't really looked into it in a while, and I don't know. I guess you can update me on the latest news.
So I think we've got good data now. That there was a paper published in February of this year that showed that older adults, they do seem to have lower levels of NAD in their muscle cells. And what's quite interesting about that paper is that there was also a correlation that older people who exercised, they seemed to have higher levels of NAD. in their muscle cells, and that correlated obviously with strength and whatnot. So I think it's going to be quite interesting to see whether if we use these NAD precursors and combine it with exercise, again similar to the rapamycin study, whether that will actually give greater muscle performance benefits compared to just exercise alone. But in terms of data that we can actually have a look at now, so I'm pretty sure these papers were published last year or it must have been last year, where they showed that the combination of nicotinamide riboside and there were 3 other supplements as well, caused a significant improvement in the recovery time from COVID-19. And I think that's quite interesting because one of the things that Dr. Charles Brenner often talks about is if we can look after our NAD levels, possibly we can make our metabolism more resilient against attacks, he calls them. So sickness is one of them, say from COVID-19, or even things like alcohol or too much sunlight. If we can make sure that our metabolism is resilient and our cells or processes don't get damaged while these attacks are happening, maybe we can get health span benefits. And only time will tell with human data whether that's true. But the COVID-19 studies, they are quite interesting because it seems to suggest that theory is correct. Now, obviously, we need a lot more data in other diseased states to know that for certain. But and one of the trials that I'm looking at quite closely is what happens if you give nicotinamide riboside or NMN to heart failure patients. So it looks like the cells from failing hearts has got significantly lowered NAD. So if we can boost that back up, what effect will that have to heart function? So I think those studies are going to be quite interesting to see what happens if we boost NAD in these diseased states. And if it proves that we should be using NAD precursors in those diseased states, should then we be using these precursors all the time anyway? Because then, say if we do go out with friends and we have a couple of drinks, should we be using NAD? I don't know what's happened to my camera there.
Go on.
Yeah, hang on. Let me switch it off, then switch it back on. Oh, sorry, team. Oh, there we go. Back. What we should be doing. Oh, sorry. I don't know what's going on there.
We can hear you fine.
Sorry, guys, we're having technical problems. This is the first time that we've done this. Yeah, I'll just use this camera instead. There we go. Can you see me now?
Yeah, we can see you.
Cool. Sorry, guys. So I think it's going to be quite interesting to see whether those precursors will actually, or whether they should be used in everyone to weather these attacks a little bit better.
Yeah, well, I was just going to add 2 points to that. One, because our cells get damaged every day, whether it's exogenous or endogenous damage. And so there's always repair processes going on within our cells. And it's possible that maybe, having higher NAD concentrations may be beneficial in enhancing that repair process, which obviously down the line would lead to less mutations. But the second thing that I think I find quite cool about NAD plus is the fact that it kind of oscillates throughout the day, like it's under circadian regulation. It's under regulation by the circadian rhythm, but it also sort of feeds back as well into the circadian rhythm. So it's involved in regulating the circadian rhythm as well. So I think so the reason I mention that is that I think there might be certain times of the day where it's more beneficial to supplement with NAD plus precursors. And so it'd be important in those studies to maybe take that into account when designing them. At least that's something I think would be important to consider. Yeah.
No, I agree. I think it's an interesting field. I think when I first started my channel, the commentary that I was hearing around NAD precursors was we could potentially use this to reverse aging, and I think we know enough now to say that that's probably not the case, particularly the data from the interventions testing program, where they trialed nicotinamide riboside, and there was no lifespan extension seen. But the idea that we might be able to use this to improve health span and weather these so-called metabolic attacks a little bit better, I think that's an interesting narrative that I'm looking forward to future trials exploring. One of the questions from chat was around Senolytics, and one of the things that I'm curious about is whether senescence actually feeds into this, into why our NAD levels lower over time. So you're obviously studying senescence, so I'm curious to hear your thoughts around senescent cells at the moment, and how they feed into our health.
Yeah, so to give some context, senescent cells, they're often described as like zombie cells, but effectively, at least the way I see it, is they're kind of damaged cells. So cells that get damaged or they've replicated too many times and their telomeres are shortened enter this process that we call cellular senescence. So there's many different ways in which a cell can become senescent, but there's like kind of general hallmarks that we can attribute to a senescent cell. One is that they stop dividing, so they've entered like a cell cycle arrest. And then the second hallmark that is very clearly present in different senescent cells is this development of a secretory phenotype that we refer to as the SASP, the senescence-associated secretory phenotype. And that includes all sorts of different molecules, a subset of them being inflammatory molecules. And so many think senescent cells as being bad because if they accumulate in the body over time, you increase the secretion of these inflammatory factors, resulting in like chronic inflammation. But I think it's like, if you understand why a cell becomes senescent in the first place, it's often very clearly a kind of tumor suppressive mechanism. You've got a cell that's become damaged, whether it's due to too much DNA damage or like, as I said, the telomeres getting too short or oxidative damage. You don't want that cell to keep on replicating because then it's, likely to form tumors. And so it's definitely a good process to be happening in the body. And it's also clearly important for early development. And so, yeah, and also wound healing. And even there is some work as well suggesting that there's the secretion of the so interleukin-6 is one of these inflammatory factors that senescent cells secrete, and that seems to actually enhance cellular reprogramming. And so it's possible that if you've got a wound and you've got some cells that have become senescent, the SASP can also signal to cells in the proximity to replicate and to kind of not only to like repair and replace tissue. So senescent cells aren't just like bad things; they also are pretty good. Does that answer your question?
Yeah, no, I think that's a really crucial point that I've also been trying to say on my channel as well, is that senescence plays a crucial role, and we probably don't want to be stopping that process. And senescent cells, they do have important functions in our body. You mentioned wound healing for one. So I think it's going to be, I think possibly there are some senescent cells that we ideally want to be clearing away, but equally there's probably some that we want to stick around. So I'm curious, there's different studies going on at the moment looking at molecules such as Fisetin to clear away senescent cells. So I'm curious, with your experience in the lab, do you think that that's a viable strategy?
Yeah, so one thing about senescent cells as well that I've kind of alluded to already is it's very heterogeneous. Like, not only are there different ways in which a cell can become senescent, also like it also seems that once a cell has become senescent, it's not like stuck in that complete state. It's kind of dynamic, and it seems that like even the secretory phenotype evolves over time, and that there might be like changes from like pro or to anti-inflammatory changes in the secretory phenotype, and that basically the big problem with the field is we don't know how to go that's a senescent cell to like the whole organism. And so that brings us back to these different senolytics, and the question is, are they targeting just a subset of these senescent cells? Are they targeting the ones that, as you say, we want to clear, or are they also affecting all senescent cells? Well, how are they impacting normal cells as well? Because these different at least the compounds that you mentioned, like Ficitin, the molecular targets are going to be present in all cell types, whether it's senescent or not. And so it must be having some kind of impact on it. It's, yeah, so I think it's hard to say for certain to what extent the clearance of the senescent cells is attributing to the beneficial Healthspan effects on the mice. Yeah, so I think there's potential, but I also think that we don't fully understand senescence enough as an entity, or like whether we should be defining subsets of senescent cells and giving them new names. I don't know, but like I think there's more to explore. And I mean, I'm biased because I'm researching the field, but yeah.
No, it is an interesting field, and I'm hoping that so the interventions testing program is trialing Ficitin in a so-called hit and run fashion. So I'm curious to see what that will show. I think it was part of the 2018 cohort of molecules that they were looking at. But also the Mayo Clinic is looking in humans at what Ficitin does. So I think it's great that these papers are going to come out and the research is being done, because I think we'll have, in a couple of years' time, I think we'll have some clearer answers as to whether we should be using molecules such as Ficitin to clear away senescent cells. One of the things that I want to ask your opinion about, though, and this has been mentioned in chat, so CD38, right? So CD38 is one of the enzymes that seems to break down NMN, and one of the potential reasons as to why NAD levels go down. And it seems that senescent cells secrete or release CD38. So I'm curious to see what your opinions are about using things like Epigenetic to reduce CD38 and to hopefully stop the NAD declining. Have you read much about that yet, or what your opinions are around CD38?
Yeah, so there's a paper that I read the end of 2020. I don't think it's per se senescent cells that secrete CD38. I think it's more the immune cells that express CD38. And so I think there was a paper that showed that the SASP, the secretory phenotype of senescent cells, increases the expression of CD38 on immune cells. And as you say, CD38 breaks down, it depletes NAD plus levels. And so that was the argument, that increased senescent cells increases CD38 in immune cells, causes decrease in NAD plus. I think that's the paper you're referring to.
Yeah, and that's right.
I think in that paper they did use Epigenetic to target CD38, but I really don't remember much more than that, and I haven't looked into it since then either.
Yeah.
Okay,
so I've just got the graph here because I've just been having a look again at CD38, because I've been using Epigenetic as a supplement to try and stop this increase in CD38 and hopefully look after my NAD levels. But obviously I'm not a molecular biologist, so I was just curious to see what your thoughts were around this strategy, because the reason why I did this video is that there was a paper published actually this month that looked at a CD38 inhibitor, and there was a lifespan extension seen in male mice. So, but again, whether this is a viable strategy or not in humans, I think only time will tell.
Yeah, that's interesting. I should probably read that. I guess it comes back to the fact that obviously CD38 is just one thing that's consuming NAD plus. Whether it's the major contributor to the decline with age, I don't know. But yeah, if it's interesting data, then I guess it's something to be interested about.
Yeah. Well, again, it's in my start, and I think that there's if we can, because I don't think we fully understand yet why there is this correlation or trend that NAD seems to go down as we age. It's I think, as Dr. Charles Brenner talks about, it could just be that over time our metabolism's getting attacked, and that over time draws from that NAD store, if you like. But equally, we've also got salvage pathways that can recycle used up NAD, if you like, and make it back into NAD. So, I think it will just be quite interesting to see if we can find ways to prevent NAD from going down in the first place, such as CD38 inhibitors, what effects that will have on human health. But again, I don't think we should get too caught up in the hype, because there's just not that much human data yet.
Yeah, for sure. I mean, it pretty much comes back to the question of what is the main drivers of aging, right? So, yeah, for sure. There's definitely so much we still don't understand.
Yeah. On the point of interesting molecules, so from the interventions testing program, we have 17-alpha estradiol, which I'm very curious to hear your opinion about, because in male mice, it was giving lifespan extension benefits. And in other trials, it seems that in male mice, it improves their muscle performance. So it sounds baffling to use a type of estrogen in males, but the data seems to be there in mice. And one of the things, just for chat, that I want to let people know about is that this is a so-called non-feminizing estrogen. But I'm very curious to hear your thoughts around 17-alpha and what the potential might be for humans.
Sure. So I don't know much more than the ITP study that you've mentioned. Obviously, it was beneficial for mice. I believe that was one of the sex-specific ones, right, where it didn't seem to have as much of an impact on females. Whether that comes down to the fact that females already have maybe higher abundance, even though it's the non-feminizing hormone, that could be one of the potential factors. I mean, just in general, sex dependent differences in aging is something that I find quite interesting, because it actually permeates down to a lot of the studies that you read. Has it just been done in male mice? So beyond just like the supplements and the ITP studies, just like even the rejuvenation studies, have they done it in both males and females? Because often you will actually see sex-dependent differences, and that's important if you're ever going to go to human translation as to something might be more effective for males, but actually females should be doing something completely different. But yeah, no, I'm sorry, I haven't actually read that much about 17 is alpha estradiol. Yeah, I don't know too much about it.
Yeah, but I think you've raised a very valid point, that the interventions that might work for females or males maybe shouldn't be used for the other gender. And I think it's going to be quite interesting to see that, because I think there's a couple of successes from the interventions testing program that seem to work in males but not females. So we've already mentioned 17-alpha, but I think one of the other ones was a combination of it's a supplement called Pretendum, which activates NRF2. That seem to give a small lifespan extension in males, but not females. So it's going to be interesting to see how that translates to humans. Because we know just from the human data that females live longer than males. So it will be interesting whether the strategies to extend male lifespan will be the same as females. Yeah, I'm curious to hear your thoughts on that.
Yeah, well, I don't know how much I can really say, but because obviously there's mouse experiments done in our research lab. I don't primarily do them, but we have recently noticed maybe something interesting going on in terms of some of the experiments. If we split up the data in terms of males and females, we see some potential interesting differences. I can't really say much more than that because I shouldn't be talking about it at all, to be honest. But definitely, I think it's something that I'm a strong advocate for, because traditionally a lot of studies were just done in male mice, and clearly there are some very fundamental differences, not only obviously in the X versus Y chromosomes, but in also just gene expression in general, and just the anatomy and how that influences not only aging but also how different interventions might manifest.
And just on the point of gender differences, so one of the things that I'm quite passionate about in the clinic is hormone replacement therapy for women. So there used to be a bit of a stigma against hormone replacement therapy or menopause hormone treatment because of some concerns around breast cancer. And it seems that at least in the human data, I think there was a landmark 2013 paper that came out, kind of dispelling those concerns for the ages of between 50 and 60. So in the women that I see who are struggling with their menopausal symptoms, one of the things that we often talk about is hormone replacement therapy, where we use a combination of estrogen and progesterone. So there also seems, and when done correctly, it also seems to strengthen or slow down the weakening of bones, and also reduce heart attack rates. So I'm the hormone space is very interesting. I'm curious to hear your thoughts around that.
Yeah, for sure. And well, so a couple of things. One, when I think about menopause, the first thing that comes to mind is osteoporosis, and obviously the fact that may be strongly correlated to the decline in estrogen that happens. I don't know. I mean, probably because I'm young, but I've not honestly looked into hormone replacement therapy for females. But obviously, another thing that's worth mentioning is, I guess, I always think there's some kind of trade-off going on to some extent between like reproduction and longevity, and how especially like, so there's been more recent data for like younger females having lower estrogen levels than they should for their age, and parts of that sometimes coming from following like so-called fad diets or not eating enough food in the first place. And I guess it kind of comes back to like caloric restriction and whether that's having potential negative consequences for things like estrogen levels in females, especially at a younger age, which is why, again, as we mentioned earlier, why I've not tried to attempt any fasting currently. So I think it's not just, it's important for health at all stages. And so I think, I think it's worth, I don't really have much more to say, but that I really hope more studies are done looking into this, especially because estrogen is one of those it's systemic molecules. It's primarily produced in the ovaries, but also it can affect many different tissues as well. Hormone signaling is quite cool.
Yeah, no, absolutely. And I think you're absolutely correct in cautioning people around some of the things that we think are confirmed. That, you know, that there are some studies showing that, actually many studies showing that actually, you know, you do worsen lifespan and health span with calorie restriction in certain genotypes. So you're right. It might be that young women should not be fasting, but we don't know that yet. So I think, it's there are things in the media, and I've been guilty on this on my channel, thinking that we've got certain so-called longevity strategies that are guaranteed, and they're really not. And classic case in point is the fasting and calorie restriction strategies. So I'm glad that you brought that up. One of the things is, just to finish up with hormones, is testosterone replacement therapy. Now, this one scares me because it's quite a famous strategy, and it's talked about from a lot of influential people. The trouble is, it's been well studied in males and older men, and it comes with significant consequences. We can see when the arteries around the heart are imaged that testosterone replacement therapy, it worsens the health of those arteries. We can see that the arteries are starting to become blocked, or that process is accelerating. We can also see that there are increases in prostate-specific antigen, which is the one of the markers that we use in clinical practice for prostate cancer. So, yeah, I just wanted to let chat be aware of that there's been a lot of human studies looking at testosterone replacement therapy, and I think caution should be the way to go with this. But yeah, I'm curious if you've looked at any particular papers or have any strong opinions about this.
So the only knowledge I have is, so I read a book the end of last year called Countdown by Dr. Shana Swan, and basically it's all, as suggested by the title, it's all about the decline in like fertility counts, and she talks about testosterone replacement therapies, and I think there's a stat in the book, or not a stat, but she wrote in the book that previously the therapy was used as a form of birth control, because if you increase the testosterone levels, it kind of signals to the brain that, oh, everything's fine, the testes are producing testosterone, we can switch off the production, and then you end up like making the whole thing worse. So at least from a fertility perspective, I don't think it's definitely something to be cautious about. Maybe short term it'd be fine, but long term, yeah, I mean, I don't know. I've not really read the studies, if I'm honest. But yeah, I don't know. Beyond fertility, I've not looked into it at all. So it's interesting to see the other consequences of it as well.
Yeah, I think I just think it's a bit scary because that's where you can, with testosterone replacement therapy, when you there are definitely instances in medicine where it should be used. Equally, it looks like from the data, if it's not used correctly, you can do significant damage to yourself. And I think, and I'm just not reading some of the comments from the influential people that talk about testosterone replacement therapy. I'm just not sure that they're aware that it's been extensively trialed in humans, and I think that that there's a reason why it's not routinely used in clinical practice, because it is associated with such significant side effects and risks. But on the point of something slightly slightly more optimistic, we've talked a little bit about SGLT2 inhibitors, where it makes the kidney pee out sugar. And why I'm quite excited about this is the interventions testing program in males, at least. I can't remember if it did it for females as well, did result in a lifespan extension. And human studies, it's used in clinical practice. And I think out of all of the molecules and supplements that I think would be used for so-called longevity purposes, I think it's the SGLT2 inhibitors that will first start to be used in clinical practice. So I'm curious what your opinions are if you've read much data around this.
Not specifically like inhibiting that protein, but just I guess in general, talking about glucose levels and high sugar diets. I'm guessing, but like so, basically the glucose signaling downstream is promoting cell growth, and it kind of comes back to everything we've sort of been talking about and how it would activate downstream proteins like mTOR, which obviously we've mentioned about rapamycin, how that's inhibiting it and appears to be beneficial. So yeah, I definitely think it seems, if it's already being used and prescribed to patients, then there's already a lot of like safety data around it. So I guess it's just about doing further testing, looking at molecular markers and other tissue types to see what other impacts it might be having, and looking where it could be prescribed beyond just for diabetes. That's as much as I know.
One of the things that I'm quite passionate about with this class of medication is that it's starting to be used in heart failure as one of them, but also chronic kidney disease. It looks like it lowers the blood or lowers the pressure in the kidney very well, and that seems to protect the kidney. So it's currently used in quite severe quite severe kidney disease. It looks like it should also be used for moderate kidney disease. And it will be interesting to see if it should be used in mild kidney disease as well. Because I think it's from the age of around 25, our kidney function starts to decline.
No, don't tell me that on my birthday. I just turned 25.
Yeah, happy birthday, by the way.
Thank you.
If we can use SGLT2 inhibitors to slow down the decline in kidney function, I think that's quite a powerful idea. And that's why I think because the human studies are coming through and they're funded, I think that's why this medication is going to be the first one that we'll use for so-called longevity purposes. So I'm out of all of the medications, I'm probably most excited for this in terms of getting it through. And then the next one's obviously rapamycin.
Yeah, I mean, just kind of speculating on from that, I guess. Obviously, so glucose alters like the osmotic potential in like the blood vessels, and so if you have higher glucose concentrations, it can, I think it, causes increased pressure because there's more water going into the blood vessels, and so like the eyes is the other thing, right? Because obviously that's quite common in diabetics, is having higher eye pressure and that decreasing vision, and so. Yeah, I don't know too much about this drug, but I guess I'll take a look at it now that you've mentioned it. It sounds interesting.
Yeah, no, I think it is. We've talked a little bit about hormone replacement therapy. One of the areas that's also getting a bit of hype at the moment is growth hormone, particularly with the TRIM trials. So the thymus regeneration, immunorestoration, and insulin mitigation trial. So to be honest, I'm not sure why this is getting why this is getting hyped, because we can see from the preclinical work that if we supplement with growth hormone, it seems to shorten lifespan. Um, but yet we've got these trials going on showing a potential thymus regeneration if we use growth hormone. So I'm I must admit I'm very wary of this trial, but I'm curious to have you done much reading about this trial, about the trim
So
trial?
yeah, so I mean that's the only trial I have read regarding the growth hormone. So that was, I think the first paper came out in 2019, where it's like a very small I think it was men only. I can't remember. But I think there was a very small number of patients. And yeah, they used DNA methylation and saw a reduction based on the methylation in the age after this combination of growth hormone, metformin, and DHEA. Is that right? I can't quite
Yeah.
remember. But yeah, I mean, yeah, I guess my initial caution is like, if you look at mouse studies where they basically mice live longer if they have either mutations in the growth hormone receptor that basically reduces signaling through this pathway. So the thought of increasing it is like, what? No. But I guess maybe there could be some way that if you could target it to specific organs or specific tissues where you want increased growth, like maybe in the thymus to regenerate, maybe it could be beneficial. But by far, I don't think there's any data that would want to make me advocate for it in any way.
Yeah, I'm kind of the same. I think it just needs to be approached with a lot of caution at this stage, and because I think there's say what would be a good example? Say nicotinamide riboside. I think there's good safety data around that. It's when used in reasonable dosages. I think it's most likely going to be safe, and so that's why I don't personally have an issue with using it. But when it comes to things like growth hormone, there is concerning preclinical data. So I certainly wouldn't want people to get the impression that they should be using or trying to source growth hormone for themselves. Um, Again, it's that caution before the hype, and I think just on that point, at least with the community here, I think a lot of people that they appreciate that we don't want to be hyping stuff because the data will speak for itself. It's not necessarily the personalities behind the data that actually matters. It's just the raw data itself, and that we don't we don't really want to be getting too carried away with things because take calorie restriction. Once upon a time, we were thinking that calorie restriction was fantastic for everyone, when actually that's probably not the case. So I do appreciate that, I can speak with someone such as yourself who is very measured with the stuff that's coming out of the lab, that, we probably shouldn't be hyping this and that and the next thing because we just simply need more data, particularly around safety.
Well, yeah, I mean, and also just on from that point, just the fact that I am a scientist and like I know the pressure and the need to publish papers and how like I have so much data that I've collected in my 2 and a bit years so far of my PhD. Is all of that going to get published? No. Like, there is some kind of, as much as a scientist you want to be unbiased, there is an element of bias in terms of what data gets selected for publication. So like how much data has been done that's not been published and could be helping us understand these different molecules or these different interventions better. That also worries me a bit. But yeah, there's no way that I also have to be careful on this platform because I've not read every paper. It would be insane if I had. I'd be very impressed with myself. But yeah, I just haven't. And I don't think anyone has, right? So you have to be careful with anyone that you listen to or to see any motivations for why they might be promoting something. So I can only, yeah, that's why, as you say, I can show the data and I can give some comments, but I can't really do much more than that because I'm the same as everyone else at the same end of the day. We're just trying to interpret what data is available and make the most rational decisions from that.
Yeah, completely agree. And from the work that you've done in your own lab, do you think one day we will be able to find out why we age and how to, in a perfect world, reverse it? Because this has kind of been bandied around for literally thousands of years that someone's got a cure for aging. So do you think
Um,
that we've actually got a chance at it?
I think we'll find. I don't think we'll ever find the actual all of the causes, or like, because one, I think there's many different potential contributors that would have different amounts that they cause to the aging process. Like, I mean, it's even like the kind of physical components, like the stress of um, oxygen pressures, or I know, like, yeah, just different environmental factors that aren't taken into account in all these different studies, as well as the internal endogenous processes that seem to malfunction with age. So I think we'll have like I'm I'm probably quite an optimistic person, but I do think we would find um, like the main causes of the aging process, and then we'll be able to use that information. Because I mean, what I was going to comment on earlier with the rejuvenation stuff we were talking about is a lot of it now is more like mechanistic, trying to interpret what's happening, what's changing in the cells in these processes, and understanding new potential molecular targets that could be more beneficial. Um, so I think, yeah, with that in mind, I think we will be able to identify certain things. I mean, obviously, this is with the knowledge that we already can do things like exercise, eat well, sleep, try to uh, not get too stressed if that's possible, will definitely help. And so, yeah, I think in general, I'm optimistic. And I mean, I want to stay in science and research, and also keen to try and help solve this problem. So I can use my brain to help solve it as well.
No, I think really well said. And on that point, because I know it's your birthday, and we've been going for an hour 20. So I really appreciate your time here, Eleanor. And chat, if you haven't checked out Eleanor's channel, I highly recommend you do. It's awesome. The animations are on point. Speaking of which, how long does it take you to do those animations?
It's very variable. It's like, so like, because obviously if you watch my videos, everything is like sped up. So it takes obviously much longer to draw it than to speed it up afterwards. But I'd actually say the hardest part for me is actually reading the papers and making sure I read it carefully and craft the script that I know is valid of the data, but also can be quite entertaining. I like to be quite funny, although I feel like I find it funnier than anyone else. But yeah, so definitely the script is the hardest part for me to work on. I actually say most of my time is spent on really making sure I read the papers carefully and I'm confident in what I'm saying, that I'm not trying to be deceptive or malinterpret data. And then actually, it's got to the point now where I really like the animations and I'm just so used to it that actually it doesn't take too long to do the drawings. And I'm also not a perfectionist, so it's a bit messy, but that's just what it is. And then, again, the editing takes a bit of time, but definitely majority of the time is reading the literature and trying to summarize that into a script. I don't know how you find it.
Yeah, no,
But
I'd
yeah.
echo that. That there's so much behind the scenes to the scripting of it, and again to make sure that you're not that you're not missing stuff. I mean, again, when I first started my channel, I was talking about molecules and essentially following certain people and following their opinions more than the data, and that led me down some pretty, uh, you know, not ideal paths. Let's put it like that. So yeah, I think double checking everything that I'm reading and making sure that getting the background around, say a new paper comes out and it's very interesting. Yes, that's great, but you also need to have a look at the background data and what everything else and what their fields have said. Because if the field has kind of started to settle on a particular conclusion, and then you've got a new paper that completely flies in the face of everything else that's come before it, then you need to interpret that with caution. So yeah, it's tricky. But I love it. I mean, I love kind of, it's almost like being a detective, that you can kind
Yeah.
of, yeah, go through all the data, try and figure it out, and then trying to find ways to actually explain it to people, and so that it's not too complicated. Because a lot of the stuff that I go through, yes, I find it interesting, but it's very, very, very detail orientated, and I personally don't include a lot of that because I don't think it brings much to the video, because I want to try and get across an overall point. So yeah, the scripting, I agree, takes a lot longer than what people would appreciate. Yeah.
Yeah. And I
But
should also
again, thank
say,
you. Go on.
congrats on your 100K. I mean, that's why we're here, right? It's very impressive. So yeah, definitely congratulations to you.
Yeah. No, thanks very much. And thanks again for coming on the show. It was an absolute blast having you.
No problem. Yeah, I'm glad that this live stream has gone well, besides your brief absence.
Yeah, besides the camera.
I was terrified for like a brief 30 seconds by myself, but
Yeah.
it's been fun.
Cool. No, thanks again. Bye.
Right. Bye.
So chat, I'm going to stick around for a little bit and answer any questions that you may have. I was kind of been keeping an eye on the different comments, and I think just during our conversation we've answered most of them. But if you've got any particular questions, let us know. So one question here from Drew is, do I still use hyaluronic acid despite studies showing it promotes cancer growth? So I think this is quite a crucial point. There's no evidence that hyaluronic acid supplements promote cancer. What seems to be happening is that cancerous cells, because of their altered processes, they seem to accelerate hyaluronic acid production. So there's a crucial difference here. The supplements, again, there's no evidence that the supplements cause cancer. It's just that the cancerous cells, through their different processes, there is that correlation with increased hyaluronic acid production. So overall, yes, I still use hyaluronic acid, and I do that because there's very good data showing improvements in skin health. So if we can improve skin health, there's a possibility that maybe it will improve blood vessel health as well. So yeah, that's why I take it. I think there's good human data around it, and again, there's no evidence that these supplements promote cancer growth. Good. Anything else? Question from here. Why do I use Fisetin only a few times a month? So with senolytic treatment, again, as Eleanor and I discussed, there still needs to be a lot more research around senolytics and how best to use them. But one of the strategies that is being promoted at the moment is this idea of hit and run. So it doesn't look like we always want to be clearing away the senescent cells. It looks like if we can have periods of time where we're trying to clear away the senescent cells, that's probably the way to go. And that's how the interventions testing program is going to be trialing Fisetin. So in those trials, the Fisetin is not used every day. I think it's used for 3 days and then a 7-day break. It's something like that. But that's why I only use Fisetin. So for 5 days in a row each month. So, for 25 days, I'm not taking the supplement. It's just for those 5 days that I take the supplement. What other questions do we have? Has Dr. Sinclair still blocked me on Twitter? Yes, he has. But again, like I alluded to before, it's not the personalities behind the data that matters. It's just the data, and I think the data speaks for itself. So I don't think that there's much point in sort of going on that phase. I just don't think it adds much. Again, at the end of the day, I think people just want to be as healthy as possible for as long as possible. And that's it. That's all that matters. So another question from chat. How do we know if any of these longevity aids really work? So this is a very crucial point. If we look at human lifespan, we're going to be waiting decades for that data to come through. So we're not going to know if these molecules actually extend lifespan in humans, because those trials, they're not feasible to run. What's interesting, so let's take my Rapamycin trial, for example. If we can use Rapamycin to improve muscle performance in older adults, that's a very powerful and it's safe. That's a very powerful reason for Rapamycin to be used in humans. So Rapamycin may improve lifespan in humans, but we just don't know that. And I highly doubt that we're going to have data to show that either way. but we might have data showing that it will improve muscle performance. So yeah, that's kind of my answer to that. That if we choose the endpoints for these trials correctly, then we can actually have a good justification to start to use some of these treatments. Question here on spermidine. So spermidine is an interesting molecule because it looks like it can promote the process of autophagy, which is clearing away old cell components. I suppose it's highly unlikely to be harmful, as in spermidine. Will it be beneficial? I just don't think that there's enough data. And the pushback that I would give to that is, if we use rapamycin, that will activate autophagy. So do we really need to use another supplement to also be activating autophagy? So I'm not personally that excited about spermidine, particularly because we can get a lot of it in our diet. So, it comes back to, great diet, regular exercise. That's what's key. So I personally don't take spermidine. I've not really seen much data to convince me otherwise. And I think we've got other molecules that are more promising. So yeah. Can I suggest one intervention for longevity? Well, if I could suggest one intervention for longevity, what would it be? This was asked by Joshua. It's the sorry to harp on about it, but diet, exercise, sleep, meditation, low stress, those are the things that actually matter for longevity and for health span. It's not necessarily the supplements. But I think what you're getting at is if I had to choose one intervention, as in one supplement or one medication, to be honest, I think it would be rapamycin. That's the shining example from the interventions testing program. So I'd probably go with that one. But again, more human data needs to come through. I don't think we should be using it at the moment. What else do we have? Someone's asking about, so Laura is asking about resveratrol. I think I've kind of made my opinion about resveratrol sort of relatively clear. I'm not convinced that there's really any benefit of taking resveratrol, and there is the suggestion that it may cause harm in terms of blunting some of the positive effects of exercise. So I personally don't take resveratrol, and I don't recommend that my patients take it either. Yeah. We've got a question here about TMG or trimethylglycine. So yes, I still take trimethylglycine, but that's primarily because I take niacin. So niacin is a molecule to boost NAD, and so it's an NAD precursor. And how niacin works is that it increases a molecule in the blood called homocysteine. So that can be a marker that we're drawing from our so-called methyl pool. And the methyl groups and methyl pool are needed for creation of neurotransmitters and all sorts of other things, DNA methylation. So I personally take TMG to so-called donate methyl groups. Is that needed? I'm not sure. I don't think we've got enough data yet. We do have data though showing that TMG, it lowers homocysteine levels. We've also got So yes, I still take TMG. When it comes to the interventions testing program, though, so they did show that glycine extends lifespan in mice. So TMG, trimethylglycine, so there is some glycine in that molecule. So for me, that's another reason to take TMG. But again, we don't have human data to prove its use yet. But at the moment, yes, I still take TMG. So I'm just going through all the questions so far. Mario is asking, how is fatherhood? To be honestly, 2021, that was the most difficult year of my life. Harry, bless him, I love my son, but he was a very difficult child. And trying to work full time at the clinic, trying to do the YouTube channel, trying to be a good father to him. That's why I get up at 4 a.m. every morning to do these videos. I got up at 3 a.m. this morning for this live stream. So, fatherhood's good, but it's very busy. But one of the things that really makes it worth it is when I come home from the clinic and I can see Harry toddle up to me with his hands outstretched, wanting hugs, and then we'll kick a ball around. So no, that's it's very rewarding. How much am I shortening? So we've got a question here about from John about shortening lifespan by running ultra marathons. So I think what you're potentially getting at here is over exercising. It seems that with exercise, you get that, you get that immediate benefit in terms of health and lifespan. And then it seems to plateau once you reach a certain amount of exercise each day. The question then is, what about over exercising? If you overstress the body, I think I'm not aware of much data around that because I don't think that there are many people that are running ultra marathons all the time. Overall, though, I until there's more data, I personally wouldn't go too much into into over exercising, because I think there is a potential of harm when people start doing that. So I think exercise every day is a good thing, but is too much. Yeah, if we overstress the body, is that a good thing? Probably not. Got a question here about telomeres. Is telomere length an indicator of longevity? Telomeres was hyped quite a bit a few years ago. Overall, I think worrying about telomere length is I don't think it's going to bear much fruit in the longevity space. I think that there are other markers of longevity that show more promise. So no, I don't think that telomere length is a particularly good indicator, and I don't think that it adds much to clinical trials when they're done. What other questions do we have? Thoughts on, so this is from Drew Dickinson, thoughts on saturated fat consumption on the keto diet relating to rising LDL levels. So this is a great question. When it comes to dietary interventions, there's actually been a lot of data showing that dietary interventions to change cholesterol levels in the blood, it's not that effective. So, there are some people that I see in the clinic who have got high LDL cholesterol, and they're eating lots of fat, lots of meat, lots of butter, and they cut that out, and they do see some improvements in their LDL levels, but it's not that much. So saturated fat content on the keto diet in terms of raising LDL, some people will have significant some people, their LDL will significantly rise on the keto diet. How detrimental is that? All the data that I've looked at seems to suggest that the lower the LDL levels, the better it is. And that's why in clinical practice, for certain patients, I prescribe them statins. Not everyone should be on statins. I don't want people to get the wrong impression here, but there are certain high-risk people that have got high levels of LDL cholesterol, and they might have high blood pressure and things. That gives good justification for starting people on statins. So when it comes to saturated fat content, I think that there's not that much evidence that it increases LDL cholesterol in a clinically meaningful way. But then the other question is saturated fat good for the body anyway? And I think the answer is probably no. How harmful is it? It's probably not that harmful, but it's not good either. I think overall, if we wanted to have a so-called healthy diet, one that's rich in whole foods, with whole vegetables and whole fruits, and a wide and varied diet is probably the way to go. And anything more than that, I'm not convinced that there's much data. So we've got a question here from Al Ali about flavonoids. So he's just bought a few bottles of astaxanthin. So astaxanthin is a molecule that activates an enzyme called NRF2, which we've also talked about on the stream so far. So NRF2 is the body's antioxidant switch, if you like. So if you can switch on NRF2, we might be able to reduce the oxidative damage or stress in our older years. And the interventions testing program will be trialing astaxanthin. And also when the interventions testing program trialed Protandim, which is a mixture of molecules that also activate NRF2, there were lifespan extension benefits seen. So I think activators of NRF2, they're very interesting. I personally use a molecule called sulforaphane to activate NRF2, but I only do that on days that I don't exercise, because when you exercise, you release all sorts of oxidants. And that actually seems to be a good thing in that short burst, because it seems that with that oxidative stress, you signal to your cells that they need to become more efficient, that the mitochondria need to deal with that stress better. So if you take away that oxidative stress from exercise, it looks like you actually blunt the positive effects of exercise. So that's why I only take sulforaphane on days that I don't exercise. So yeah, that in terms of astaxanthin, I'm very curious to see what the interventions testing program says. We've got a question about my shiny glowing skin. So it's just the lights that I've got here. And that's essentially it. So we've got a question here about rapamycin and how it's a prescription medication. So how will regular people access it in the future? So what I'm hoping with the clinical trials that I'm running, I'm hoping that if we can show that rapamycin is safe and that it improves muscle performance, that gives a strong justification for doctors such as myself to be routinely prescribing rapamycin. So we can actually get it cleared by the FDA. So I'm hoping that way we can almost open up the floodgates on rapamycin. Now, again, that all depends on what the data shows in these clinical trials. I'm hopeful, but equally, we might find that rapamycin doesn't improve muscle performance in older adults. I don't think that will be the case, but the data will show. So to answer your question, I'm hoping that as more trials come about and we can start to see benefits of rapamycin, it will become commonplace for medical doctors such as myself to be prescribing rapamycin. What other questions do we have here? I hope so. We've got a question here about changing my opinions around metformin. So again, my opinion literally doesn't matter. It's what the data shows. So I think there are 3 crucial trials when it comes to metformin. The first one is the interventions testing program. When they trialed metformin, there was no lifespan extension seen. So that's a crucial finding. The second study was published in the journal Aging Cell in 2019. And this was a study that looked at non-diabetics and it wanted to see what was the effect of metformin on exercise. We've got very good human data showing that metformin blunts the positive effects of exercise. It was by around 50%, and this was measured when the participants were, I think they were on an exercycle, and they were measuring VO2 max. So it's very good human data showing that metformin does blunt the positive effects of exercise. So that's the second trial. The third trial, there was a long-term placebo-controlled study in humans. It started recruiting patients at the end of the 1990s. And what it wanted to look at is, could you use metformin in non-diabetics who were overweight or at risk of developing type 2 diabetes? Could you use metformin as a preventative strategy to hopefully reduce the chance of developing new diabetes and hopefully extend lifespan? In that trial, there was no benefit seen in terms of cancer, in terms of heart disease, or in terms of lifespan. That was a long-term, well-powered study. So, with that combination of studies, I don't see a strong reason to be using metformin in non-diabetic people. When it comes to diabetics, that's a completely different kettle of fish. So if I see a patient in the clinic and I can see that they are pre-diabetic, I will suggest to them that they should be on metformin. If they are a type 2 diabetic, metformin is the first one that we go for, obviously alongside a good diet and regular exercise. So, in type 2 diabetics and pre-diabetics, yes, metformin absolutely makes sense. When it comes to non-diabetics, I think we've got good data to show that's probably not a great idea. What other questions do we have here? We've got a question about excessive protein intake, whether that will shorten lifespan and health span. This is a great question. There's a interesting dynamic with protein. Now, it looks like if you restrict protein in mice, in some mice species, you probably increase lifespan. But if you restrict protein, you're probably going to make that organism weaker. So you've got this interesting trade-off because, again, when you get older, you want to make sure that you're holding on to your muscle stores. Because like we've gone through in this stream, I see so many patients in the clinic who, unfortunately, they've lost their muscle strength. They are dependent on their carers. They have to go into rest homes and private hospitals. So we want to prevent that happening. So if you're restricting protein, there's the potential that actually we could be doing harm because the muscle wasting process will accelerate. So for me, I just try and maintain a generally healthy level of protein, not too much, not too little. And it's just that balance that I'm trying to strike between lifespan and also making sure that those years that I'm on planet Earth are as strong as possible. So yeah, that's where I'm at with protein. And for my older people, I often suggest to them to increase their protein intake because when I'm asking them about their protein, they often don't have much in their diet. And that's to be probably the last thing that we want in those older years. We probably want to be increasing the protein intake, again, to hold on to that muscle strength as they age. What else do we have? Thoughts on Sirtuin 6 activators. So the data that we've got so far on this is in genetically modified mice. Those mice were genetically modified to overexpress Sirtuin 6 or Sirt6. And with those mice, there was a lifespan extension seen. So that's very interesting. But obviously the caveats to that is that, first of all, they're mice and they're genetically altered. So we don't have, to the best of my knowledge, we don't have any data showing that a supplement which activates Sirtuin 6, what that does to the mice, whether it extends lifespan or improves aspects of health. So I'm interested and I'm definitely following this data, but would I spend money on buying a so-called Sirtuin 6 activator? I don't want to spend my money on that personally. I just don't think that there's enough data yet. But it's definitely an area that I'm following closely. Other questions do we have that we haven't answered yet? Yeah, anyone else have any other questions before we wrap things up? We've from Rose, she's mentioned collagen. So collagen is a supplement that I take. I mix into my morning coffee every day. And if I'm having some porridge, I also mix in collagen. So when I first looked at collagen, I thought it was just the biggest load of baloney. But when you have a look at the data, there's good human data showing that collagen supplements, they do improve skin health. They do reduce wrinkles. And based upon that data, I think that's a good reason to use collagen. Personally, there doesn't seem to be any downside aside from cost. But if we can see aspects of our skin improving, will that improve other aspects? Will that improve blood vessel health? Will that improve the cartilage in our joints? Who knows? And I think time will tell on that. But also, and on this point, if you're looking in the mirror and you can see that, and you're feeling young, a lot of being young is how you're feeling. So if you're feeling young, you can almost trick your body into saying, yes, you are young, and improve your health that way. So I think people sometimes don't appreciate that the positive effects that skin has on our general health. That, again, if we're looking in the mirror and we see a young person, we're going to act like a young person. Just subconsciously, we're going to have a bit more motivation, maybe to do, to make good diet choices and to go on that run. So yeah, I think collagen is a very interesting supplement that I personally take. Question from Patrick: why do females live longer? So females generally have much lower rates of cardiovascular disease. Um, and testosterone may have a role to play in that. Estrogen also may have a protective role in that. I don't think I'm not aware of the exact mechanisms as to why females generally live longer than males, but the data is quite clear there. Um, so yeah, I don't have a great answer for you, but it's probably to do with the different hormone levels and the effects that has on our body. So again, that's one of the big reasons why I'm very excited to read data from the interventions testing program, where let's take SGLT2 inhibitors. That seems to give a lifespan extension in males. Um, so I think we're going to find ways to potentially bridge that gap, but hopefully with other molecules such as rapamycin, we can get lifespan extension or health benefits in both genders. And that's why I've chosen to study rapamycin in my clinical trial. We've got a question from Eric. Is calcium alpha ketoglutarate better than NMN? So uh, with this question, I'm not convinced that I can answer this. I don't think that there's a better molecule, because they work on different pathways. Um, I think that the evidence around NMN and other NAD precursors is interesting, and I'm excited to read more, specifically in diseased states like the heart disease that we've mentioned before, or COVID-19. Those are quite interesting. When it comes to calcium alpha ketoglutarate, there's an interesting mice paper. I think it came out in 2020 that showed a lifespan extension, and that was from Dr. Brian Kennedy's lab. So calcium alpha ketoglutarate, it may have a important role to play in humans as well. I'm just not aware of much human data yet showing improvements in any functional outcomes. It may be that alpha ketoglutarate improves muscle performance, in which case that's a great reason to start taking it. But at this stage, I personally haven't come across any convincing human data yet for me to start taking it. Got a question from Danny. What is your opinion about omega-3 sources? So it's a great question, because whenever considering a supplement, we have to make sure it's safe. And that also includes the additives that are actually in the supplement. So in a perfect world, we want to make sure that the supplements, if we choose to take any, are third-party tested. So there are different third-party testing outfits. So one of them is called LabDoor. So I have a look at LabDoor whenever I'm choosing a supplement, and on there they third-party test them, and you can figure out, is what's actually on the label what you're taking in that supplement? Are there any worrying things within that supplement that you ideally don't want to take? So using websites such as LabDoor, that's where I source my my omega-3 brand from, and I do that for things like creatine and whatnot as well. So I hope that's answered that question. So we've got a question from Ryan and Stacy. We've got a question about probiotics and their impact on longevity. So the gut microbiome that has a crucial role to play in our health. And we alluded to this a little bit earlier on in the stream when it comes to weight loss data. So if we don't feed the gut microbiome or bacteria, they have specific interactions with our body that cause us to feel very hungry. And that's one of the reasons why, if we supplement or have high fiber foods, that feeds our gut bacteria really well. And it can almost help to trick our bodies into thinking that we're full. That's only one thing that the gut bacteria does, and there's a myriad of other things that the gut bacteria has, and lots of different interactions with our immune system. So the question then is, what should we do about it? Because there's many different strains of bacteria that live within us. So how do we know which strain of bacteria will actually give us health benefits and which ones we should try to avoid? We don't know. There's a lot of data that's been sponsored by probiotic supplement companies showing benefits. I'm not aware of much human data that's not sponsored by these companies that show a benefit. I think with the age of so-called big data, we might be able to find the answers as to which bacteria we actually want to be promoting their growth and how best to do that. But in the meantime, I personally just want to focus um, on having lots of fiber in the diet and having a wide and varied diet, because it seems that by having a wide and varied diet, you have lots of different types of gut bacteria, and that seems to be a good thing. So that's currently my strategy, and I'm eagerly awaiting more data to come through. But at this point, I do not suggest to my patients that they should spend money on probiotics. So we've got a question from Mario. About hyaluronic acid. Is it better taken topically, as in a cream, or ingested? So ideally, what we want is to promote the health of the skin at the deeper layers, because how the skin works is that it's constantly refreshing itself. So from the bottom, you'll have new skin cells, and they will work their way up to the outside world and then eventually shed away. So ideally, you want to be focusing on that lower, on the lower layers, where you can actually make a significant difference to your skin health. So that's why personally, I use a supplement. I don't use a hyaluronic acid cream. Question from Jay Miller about creatine. Is there any reason to stop or decrease the intake of creatine? I've heard this quite a bit in the comment section. I'm not aware of much human data, and please chat, correct me if I'm wrong, of so-called pulsing creatine. I take creatine every day. So I take it on the days that I work out as well as the days that I don't. And I've not seen much data to suggest that stopping creatine for a period of time is beneficial. And I'm not. I think it would be quite difficult to get funding for that type of study as well. So yeah, I'm personally don't pulse it. So we've got a question about rapamycin as an immunosuppressant. So yeah, we went through this earlier on in the stream. One of the concerns about using rapamycin is, will we negatively influence the immune system? As in, if we start taking rapamycin, will we be more susceptible to infections? And will we slow down wound healing? I think those are important questions. And in my mind, I think we've already answered them. We've got a human study, I think it was of 268 humans, placebo-controlled, that looked at the influenza infection rate and the response to the influenza vaccine. Those that were taking mTOR inhibitors, which is what rapamycin is, there was an improved immune response. So on the back of that data, I'm not particularly concerned about the so-called immunosuppressive effects, because I don't think that if we take rapamycin once a week, I don't think that we would see those effects. And if anything, I think we'd probably see an improvement in our immune system, as that trial showed. So it's definitely something, when we're running these clinical trials, that we need to be aware of. That should be a safety consideration. But I'm not particularly concerned about it based on that large-scale trial that we've done. Drew also is asking about, where did I learn to become a YouTuber? So I follow a couple of finance channels, and one of the finance channels that I follow is Graham Stephen, and he has a YouTube course about how to be a YouTuber, and I started that. So I mean, when you look at my initial videos, they are terrible. They are so bad, and I think I've come a long way since then. And even just being a bit more relaxed in front of the camera, I'm certainly not a born showman, and I definitely wouldn't say that I'm a showman now. I think I've still got a long way to go. So I started this channel because I realized that there was so many people talking about health, and what they were doing is often extrapolating cell or mice data, and trying to make that apply to humans. And I wanted to try and fix that, where I could actually present the human data to people and say, this is why I'm doing X, Y, and Z to improve health. So I just wanted to create a resource that people could go to, where they could see the research for themselves and make their own decisions. And so far, with the I think we've managed to get a pretty awesome community who enjoys that, who want to see the research for themselves and want to get away from the hype of different supplements and so-called fads. So yeah, that's how I learned to be a researcher, a YouTuber, and I'm still learning. But that's one of the exciting things. And I think if you, one of the pieces of advice that I did want to give people is that if you ever thought about starting a YouTube channel, just start. Like you'll get better. Have a look at any of my older videos, and they are so cringe. I don't understand how I got any subscribers from them, because if I went onto those videos, I would immediately click away. So it's just time, and you just got to start somewhere. So yeah, do it. So we've got a question from Patrick Steele. Would I avoid taking broccoli sprouts on workout days? So I think what you're alluding to, broccoli sprouts, if you take them, they're a fantastic way of naturally creating sulforaphane, and sulforaphane activates NRF2, so the master antioxidant for the body. So like we've gone through, it seems that if you want to have that initial oxidant stress in response to exercise, so that your body can respond to that oxidative stress. So the question is, if we take broccoli sprouts, will we also blunt those effects of exercise? I'm not aware of any human data that would suggest that's a bad thing. I think you'd have to be eating a lot of broccoli sprouts to cause negative impacts. Overall, if people want to eat broccoli sprouts, I think that's great, and I wouldn't want to discourage that. So I don't think at this stage that there's really much data to suggest that we shouldn't be eating broccoli sprouts on workout days. If anything, if more people could be eating broccoli sprouts, I think that's great. Any other questions? We've got a question from Jay around olive oil and activating sirtuin. So extra virgin olive oil is probably a good thing. That if people want to use it on salads, it's probably a good thing. How it exactly works, I'm not sure. Does it work by activating sirtuins? Does it work by activating a whole lot of other things? Does it interact with the gut microbiome? Not quite sure, but overall, extra virgin olive oil appears to be a good thing to include in the diet. So when I have salads at night, we always drizzle a little bit of extra virgin olive oil. What else do we have? Estrogen for longevity. So we've talked a little bit about this on the channel, where we mentioned 17-alpha estradiol, which is a type of estrogen. So this is an estrogen that it's non-feminizing. So when the male mice took this particular form of estrogen, it wasn't causing feminizing effects. What it did seem to do is extend lifespan, and also it seems to improve muscle performance in those mice. So I'm very interested to see whether that works out in humans. And again, I would want to be doing an exercise trial with 17-alpha to see if we could get muscle performance benefits in older men if they took 17-alpha estradiol. I think that would be a fascinating study to do. To be honest, I just wish that I wish that I had more access to funding. There are so many trials that I'd love to run, and so many interesting molecules. So 17-alpha is definitely up there. In terms of estrogen for women, so like I've kind of gone through, I'm often prescribing estrogen to my women aged between sort of 48 to 60 to help with their menopausal symptoms. And there's many reasons for that. It seems, so yes, to help with the symptoms, and that's a crucial point. It seems to lower cardiovascular disease or heart disease. And it also seems to improve bone health, as in it seems to slow down the weakening of the bones as we get older. So I think there's a lot of different reasons to be prescribing estrogen. So yeah, it'll be interesting to see, particularly with males, whether using non-feminizing estrogen such as 17-alpha is a good idea or not. What else do we have? David is asking a really good question. Is there a supplement that I'm currently taking that I'm beginning to question whether it's truly necessary? It's interesting that you say that. I was on the cusp of stopping Epigenetic. So Epigenetic, we've talked about already on the stream, where it seems to block an enzyme called CD38. And the preclinical work is interesting, but really there's no human data really that shows that's been done on Epigenetic. So I was about to stop it, but then a mice trial came out that, for me, suggested that there probably would be benefits of taking Epigenetic. So yeah, so wait for that video. But that was definitely one of the supplements that I was about to stop taking. But I'll continue. Other supplements that I'm not convinced about. Again, we are waiting for more human data to come through, so that this will always be subject to change. And I'll let you guys know if anything changes. But for me, no, I'm pretty proud of the supplements that I currently take. Talking with other researchers such as Dr. Matt Kaberlein, Dr. Morgan Levine, so high-powered scientists who take virtually no supplements. Dr. Morgan Levine takes no supplements, and Dr. Matt Kaberlein only takes rapamycin. So that is food for thought. That if those scientists aren't taking any supplements, I think that speaks volumes. That really it's diet, exercise, sleep, meditation. Those are the things that really matter. But for me, with the data that I've personally read, I'm pretty comfortable and happy with the supplements that I'm currently taking. Merlin is asking about the antioxidant pterostilbene. So pterostilbene is the methylated version of resveratrol. And for me, pterostilbene and resveratrol, they sit kind of in the same basket. That the preclinical work doesn't show benefit, and the human data doesn't show any benefit either. So personally, I don't take pterostilbene and I don't take resveratrol. Any other questions that we've got, team? Lance Carter is asking about berberine. So if metformin isn't so good, what about berberine? So berberine is a weak activator of an enzyme called AMPK, and that's one of the Sorry, guys. Not too sure. No, sorry. Technical problems. First time doing the stream, so apologies. If we can go back to the questions. Sorry about all that. Yeah, so we were talking about berberine. Sorry, guys. Since berberine and since berberine also activates AMPK, but it's a weak activator of it, I'm not particularly convinced that that's going to do much good for us. When it comes to the other effects that berberine may have, so there's another class of cholesterol medications that's coming through, and berberine looks to be similar to that class of medication. So maybe berberine will have positive effects on our cholesterol levels, but again, I think that the evidence is quite weak. So personally, I've not seen much reason to take berberine. Again, sorry for thank you for the super chat, Patrick Steele. Really appreciate that. Any other questions that we've got? We've got a question here asking, when will we stop or reverse aging? I think we're going to find ways to extend health span. I think we're going to find ways, possibly through rapamycin, to improve muscle performance in older adults. In terms of being able to take a 80-year-old and turn them into a 60-year-old, if we're going to have, if that is going to bear fruit, I think it's going to be through the epigenetic reprogramming. But again, chatting with Dr. Morgan Levine, I think we're a number of years away from that, if ever. I think that research is exciting, but yeah, I think we've still got a long way to go. People have been saying that they've got the cure for aging for literally thousands of years, and I think now we're entering a phase where we may actually be able to have a significant impact on aging, because of the technology that we've got available and the scientists that we've got available. But I think we're a long way away from being able to reverse aging at this point, unfortunately. Cool. Any other questions that we haven't already answered? Cold therapy. This is interesting. So this kind of speaks to the idea that stressing our body might be a good thing to do. So stressing our body with exercise seems to be fantastic. If I could put exercise into a pill and give it to all of my patients, I absolutely would. So then the question is, could we pair exercise with other forms of stress? So some of the interesting papers around this came from Finland, where they looked at Finnish saunas, and they wanted to see if you combine saunas with exercise, would that give greater benefits for cardiovascular disease or heart disease? And it seemed from that observational data that yes, if you combine exercise with a sauna bath, you seem to get even greater benefits, which is fantastic. Again, that's observational data, so it still needs more work. But what about if you use cold therapy? I'm not aware of much human data on this, particularly over the long term. But the idea of stressing our bodies in a meaningful way seems to bear fruit. So if people want to do ice water baths, great, so long as they do it safely. But I think the jury is still out as to whether cold therapy will give benefits. So personally, I don't do ice water baths, but maybe in the future with more data, maybe it will come through. Welcome back. Yeah, sorry guys. Is any kind of stress good? So Paul Wolf is asking this. Absolutely. So exercise is the number one stressor. Again, if we could bottle that up, absolutely. So, I can't oversell exercise enough. If all of my patients could exercise half an hour a day, I think I'd be seeing far less patients in the clinic. What else have we got? Cool. So we've got a question about fasting, about the 18-6. So this is time-restricted feeding, where you're only eating within a small 8-hour window in the day, and fasting for the other 16. This developed a lot of excitement because if you starve a mouse for 16 hours, we seem to get lifespan extension. We also seem to get, reduced levels of diabetes and of cancer. So the hope was that if humans also fast for 16 hours, will that also give those benefits? And so far, it seems that the answer is no. That if we calorie match the different groups, so if one group is doing the time restricted feeding, where they're only eating within an 8-hour window in the day, and the other group is just eating whenever they want. But if they're eating the same amount of calories, it seems that you don't get any extra benefits. And there was a Cochrane review published in 2021 that confirmed that. So I personally don't do time restricted feeding because I'm not convinced that the data is there to support it. I think a mouse fasting for 16 hours is incredibly different to a human fasting for 16 hours. So that's why I do prolonged fasts roughly every 3 months, because that to me has got a mechanism for how it may improve health. But like we were also going through with Eleanor, it's possible that maybe only a certain selection of the population should be doing these fasts. It might be that young women shouldn't be fasting, and more work needs to be done around that. So that's a crucial safety point. That while things may look promising in the mice world, that doesn't always mean that it's going to translate into humans. Yeah, Mark Lloyd is asking, do I take Fisetin and quercetin at the same time to boost their senolytic activity? So yes, I take both of those supplements for 5 days in a row each month. And again, I'm excited about the preclinical work, but there's not that much clinical data in humans to show health benefits of Fisetin and quercetin. The Mayo Clinic is trying to fix that because they're running some trials looking at Fisetin in that so-called hit and run fashion. And I'm very excited to read that data. So yeah, we'll see. So we've got another question from Danny. Should vitamin D be paired with magnesium? So I take both of those supplements every day. So I think that the data specifically around vitamin D is quite compelling now. Specifically, I did a video on this recently showing a 22% reduction in the development of new autoimmune diseases when patients took vitamin D compared to placebo. So for me, that's a powerful reason to be talking about vitamin D with my patients, specifically because a lot of us, we don't get enough vitamin D. We are indoors working in offices. So like any vitamin, you want to make sure that you're getting enough of it. So if we're not seeing enough sunlight, we're not going to get enough vitamin D. So it seems to me to be a no-brainer to take a small dose of vitamin D each day. And that's what I talk about with my patients. And again, with magnesium, I take that at night every day. So we've got a question. If I was living on a deserted island and could only take 2 supplements, what would they be? Food and water. But I think I know what you're getting at, is that if I had to spend money on 2 supplements, what would they be? And at the moment, I'd probably say omega-3 and vitamin D, in addition to a great diet and regular exercise. It would probably be those 2. We've got a question from Ryan and Stacy. Coffee. I love coffee. Is it aging me? So there's been a lot of data looking at coffee, and thankfully, because I'm a bit addicted to coffee as well, coffee, if anything, it actually causes health benefits. It looks like it lowers heart disease, which is interesting. Now this is observational data, so looking in the rearview mirror. So it might be that people who have coffee every day, it may be that maybe they're just wealthier, that they can actually afford to have coffee every day, and that's why they've got lower heart disease. When the authors of these papers try and control for those factors, though, it looks like people who do drink coffee do get these benefits. So personally, I have 2 coffees every day. but I make sure to not have coffee beyond about 9 or 10 a.m. because the caffeine in the coffee, it sticks around in the body for quite some time, and that does impact your sleep. So that's one thing that if patients are asking me about coffee, I would recommend to them, don't have coffee after 9 or 10 a.m. But overall, it seems that coffee is a good thing. We've got a question from Joshua. Does sunscreen increase longevity by protecting against UV-induced DNA damage? So every morning I put sunscreen on my face. And there's many reasons for that. But primarily it's to protect my skin against UV damage. So it comes back to that thing that we talked about earlier on in this chat, on this live stream. If you look in the mirror and you look at yourself and you're like, you know what, I look young and I feel young, you can trick your body into actually being young. It gives you a bit more motivation to look after your body, to exercise, to eat well. So it's not just vanity for me with trying to look after the skin. For me, you can trick yourself into thinking that, yes, I am young, I want to remain young, and I want to do everything in my power to be young. So yeah, I put sunscreen on every morning to protect my skin. So we've got an interesting question from Joe Santos asking if stressing the body is a good thing. Why depriving sleep? Why is depriving sleep not good? So this comes back to what type of stress do we want to put the body under? Exercise seems to be a fantastic type of stress. Other sorts of stresses, probably not. So let's say if we've got a particularly stressful job, that type of stress is not good for us. If we are sleep deprived, that type of stress is not good. If we're interrupting our circadian rhythm, so shift workers, that type of stress is terrible for the body. The data is quite conclusive on that. So it just depends on what type of stress we're actually putting the body under. There are some stresses that are great. There are other stresses that are really not. Got a question about, can I comment on glycine and NAC? So these are both molecules that I plan to start taking when I reach the age of 50 years old. And the reason for that, one of the most powerful antioxidants that we've got in the body is glutathione. And there's good human data showing that glutathione levels in the blood go down from the age of around 45. Up until then, it's relatively constant. So it seems that once we reach that sort of 45 or 50 year mark, the antioxidants, they start winning. And that's not what we want. We want to try and maintain that balance, it seems, where we don't want the oxidants to run away on us. But equally, we don't want the antioxidants to run away on us either. So that's why at the moment I don't take those supplements. Again, I take trimethylglycine, but that works slightly differently to just taking glycine by itself. Any other great question here about liposomal NAD. So, or liposomal NMN. So, I think it's really important because liposomal supplements, they are taking off. You can get a lot of things that are in their so-called liposomal form. So it's important to actually go through what is a liposomal supplement. Essentially, what the drug or supplement companies are doing is that they're taking that supplement and then they're wrapping it in a protective layer, if you like. And that protective layer means that your body can actually absorb that supplement and then it can actually get directly into your cells. So let's take NAD, for example. There was a study published, I think it was in 2018, showing that nicotinamide riboside over a 3-month period did not increase muscle NAD levels. So then the question is, well, why is that? And maybe it's just that the supplement wasn't getting to the muscle in a meaningful way. So that led some companies to create liposomal forms to see if we could get that supplement to where we want it to go to actually have an improvement in health. So it's an interesting area of research. My concern is that so let's take NMN. If you package NMN up in a liposomal form, that NMN can directly enter the cell. There's There's nothing that the cell can do to stop that from happening. So there's the theoretical risk that if you have too much NMN, you will activate a cell death pathway. So if you, through the liposomal NMN, if you're directly piling all of this NMN into the cell, that can potentially be a very bad thing. So personally, I'm staying well away from liposomal supplements at this stage because there's not enough safety data yet. Again, there's no way for the body to control whatever that supplement is from getting to wherever it can go. So you can just flood your cells with all of these supplements, and that might not be a good thing. And there's no way for your body to regulate and control that. So until there's safety data, I'm staying well away from liposomal supplements. Cool. Any other questions that we haven't already gone through in the stream, guys? I'm going to have to shoot off to the clinic soon, but I can probably squeeze in a couple more questions. Oh, we've got a question again from Mark Lloyd. So it's around hyaluronic acid again. So there are different forms of hyaluronic acid. You've got short chain and long chain, or low molecular weight and high molecular weight hyaluronic acid. The one that I personally take is high molecular weight. There's not a lot of human data that I'm aware of that will say that one's better than the other. But it seems that in the naked mole rat, which is one of the organisms as to why we started to get excited about hyaluronic acid, it was the high molecular weight hyaluronic acid that seems to be one of the reasons as to why the naked mole rat has got such significant lifespan compared to others in its compared to other species. So yeah, high molecular weight is what I'm currently taking. We've got a question from Rose about no flush niacin. So if you actually have a look at no flush niacin, what it typically is nicotinamide. So nicotinamide can also boost NAD levels. There's also human data showing that in people that have had previous skin cancers, that taking nicotinamide seems to reduce the chance of developing new skin cancers, which is interesting. Nicotinamide, though, so once NAD is used up by a particular enzyme, nicotinamide is created. And yes, that nicotinamide can then be recycled into NAD again, but it seems that ideally we want to take a precursor that is directly transformed into NAD as opposed to the byproduct of NAD. So yeah, I don't take nicotinamide, and I don't take no flush niacin. Yeah, it's a bit theoretical, to be honest. There's not a lot of human data showing that one precursor. Well, yeah, there's not a lot of human data showing that one precursor is better than another. But yeah, I don't take niacin. Sorry, I don't take no flush niacin. Drew again is asking a question about vitamin D excess. It's a question about risk. Too much vitamin D has the theoretical potential of causing high levels of calcium buildup and kidney stones. That's the theoretical risk if we overdose vitamin D. There are some studies coming out. So there's good data now showing that taking 4,000 units of vitamin D seems to be a safe thing to do over the long term. That was a well-powered study. But taking more than that, what benefit is there going to be? Again, this vitamin D, it's just a vitamin. All we want to do is just make sure that we're getting enough of it. And having these super doses of vitamin D, there's no human data showing that that's a good thing to do. And you're potentially exposing yourself to the negative aspects of it. So I would not advocate at all to be super dosing vitamin D. I think between 2,000 and 4,000 units every day is probably safe, probably gives you benefits. Anything more than that, And I think you start to get diminishing returns and you start to increase the chance of developing side effects. Paul is asking what I would recommend for people over 50 years old. So in each video, I've got a link to my supplement stack. And in that supplement stack, I say what I would do if I was 50 years or older. John is asking, if there's any advice for someone who wants to become a physician? I think you just have to really know that you want it. It's a tough road, and it's a very tough road. So I think just know that you want it. And particularly say in the States, it's very expensive, and you'll be paying off student debt for a long period of time. So yeah, we need physicians, obviously, but it's certainly not without its sacrifices. It's very rewarding. Like, I'm potentially helping people here on this live stream. I'm about to go to the clinic and help people face to face. So it's very rewarding. But yeah, just make sure that you know that you want it. Cool. All right, guys. I think I'm going to have to finish up. I need to go and get my son out of bed, and I need to shoot off to the clinic. But hey, this was a lot of fun. Sorry for the technical difficulties with my camera bugging out and then leaving the stream for a little while. But no, I had a lot of fun. Thank you for all of the comments, and thank you so much for your support. I mean, the fact that we've got to 100,000 subscribers is it's unreal. So I'm just so grateful that that there's that many people that actually want to tune in to the research that we go through. So I really hope that you guys get a lot of benefit from these videos, because you all choose to give up some of your time to watch what I have to say, and I'm honored, and I just hope that I'm rewarding your decision as best as I can. So again, thank you so much. Thank you so much. And I think I want to do these live streams maybe once every quarter, sort of every 3 months, to answer any questions that you've got. But yeah, no, a lot of fun. So thanks, everyone. All the best.